Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
A T cell, upper left, attached to a cancer cell, lower right, by a nanotube at Tanmoy Saha. Cancer cells send out nanotubes to suck mitochondria from immune cells, finds a November 18 study in Nature Nanotechnology. It's surprising that the transfer of mitochondria happened between different cell types, intriguingly between immune cells and cancer cells, writes cancer biologist Ming Tan of China Medical University in Taiwan, who was not involved in this study, in an email to the scientist. While researchers have observed mitochondrial transfer between cells before, most cases occurred between two cells of the same type. Moreover, the mitochondrial transfer appears to have a significant impact on tumor cells escaping from immune surveillance, Tan adds. This is exciting because, of, its potential therapeutic implications. See, nanotubes link immune cells. Shiladitya Sengupta and colleagues at Brigham and Women's Hospital and MIT, including co-corresponding author He-Lin Zhang, stumbled across the behavior of these nanotubes as they were taking a. What we are trying to do is build a model system to study how cancer cells and immune cells interact using tools from nanotechnology, going into resolutions not possible with light microscopy. Mitochondria, green, traveling from a T cell toward a cancer cell through a nanotube, red. To determine what cargo passes through the tubes, Sengupta and colleagues labeled mitochondria with mitotracker green and amine-containing molecules in the cytoplasm with cell trace far red. Clearly, the fact that mitochondrial trafficking is faster means that it is an active process that is happening. The team also found that cancer cells grown in culture with T cells consumed about twice as much oxygen as the same cells grown by themselves, indicating that mitochondria pilfered Fr. is very remarkable, says oncologist Emil Liu of the University of Minnesota Medical School, who was not involved in the study. This study opens up the possibility that the cancer cells are. It would be interesting to know what other organelles or other cellular materials beside mitochondria can be transferred through nanotubes, and what their impact on cancer cells and immune cells may be. See cancerous conduits. Currently, no specific inhibitors are available to block the forma. So, to test how inhibiting mitochondrial transfer affects cancer cells, the researchers inhibited ROS/Rho-GT-PACE signaling instead, which provides the fuel for the formation of nanotubes. When the researchers injected the inhibitor alone into mouse models of human breast cancer, however, tumor growth was not reduced. The scientists did observe a reduction in growth when the ROS/Rho-GT-PACE inhibitor was combined with a PD-1 inhibitor, an immunotherapy drug that blocks cancer cells from silencing immune cells. If I can prevent that, I'd get more immune cells in the tumor, and that's the goal. Sengupta writes in an email that he and his colleagues at Brigham and Women's are developing more specific inhibitors of nanotube formation, and have filed a patent. Occurred because of nanotubes is difficult to say based on the data provided, he says, adding that although he regards the study as very in-depth, it lacked compelling, in vivo evidence of nanotubes forming in tumors. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.